Hello everyone! Today we will explain the circulation process in 5 key steps of metastasis. Without further ado, let's go. Before we start, as we all know, formation of metastasis has 5 keys, and we already learned about invasion, intravasation, and now we're going to learn about circulation. So let's start. As introduction, we're using term circulating tumor cell or CTC. CTC is the cancer cell that shed from the primary tumor and enter the circulatory system. There are 2 pathways for CTC metastasis in the circulation, which are through blood vessels and through lymphatic vessels. So let's move to the first one. Metastasis through blood vessels. CTCs are shed from primary tumor sites and travel through the bloodstream, mostly as single CTCs, but some can be detected as CTC clusters or CTC neutrophil clusters. But there is a high probability that the tumor cells will be destroyed due to the immune response of the body, so in order to survive, the CTCs will stick together with platelet, macrophage, or neutrophils. So we go to the first way of survival, which is the interaction between CTCs and neutrophils. Neutrophils are a component of the innate immune system and the most abundant white blood cell, WBC, in the human circulation. But when neutrophils interact with CTCs, it can influence the metastatic potential of cancer cells and contribute to the formation of a microenvironment that supports cancer cell survival and spread. So how does neutrophil help CTCs? First thing first, CTCs will release chemotactic signals such as chemokines and interleukin-8. Chemotactic signals are chemical signals that make neutrophils move towards a source of the signaling. Neutrophils are moved to the site of the tumor through the bloodstream, guided by these chemotactic signals. Next, the neutrophil will release neutrophil extracellular traps, nets. Nets are web-like structures composed of DNA, histones, and proteins. These nets have the ability to capture circulating cancer cells, CTCs, and CTCs, adhere to nets through a specific protein called beta-1, integrin. Moreover, nets can wake up dormant cancer cells and facilitate metastasis. Nets also can suppress the activation of various immune cells, including natural killer NK cells, which play a role in fighting tumors. Next, CTCs also will induce changes in neutrophils. Tumor cells will induce phenotypic changes in neutrophils, altering their function and behavior. These changes may include the changes of immunosuppressive properties, which could create an environment conducive to tumor progression. Next is the interaction of CTCs with platelets. When CTCs enter circulation, CTCs immediately come into contact with platelets and induce their activation and aggregation. This process is known as tumor cell-induced platelet aggregation, TCIPA. Following the activation of platelets, the platelets will accumulate on the surface of CTCs to form a shield that protects CTCs from the destruction by shear stress and immune recognition. Besides that, the platelet-CTC interaction also can lead to the transfer of platelet MHCY to tumor cells. So when this occurs, it will prevent the identification of CTC and help the CTCs to escape from the cytolytic activity mediated by NK cells. Last but not least, upon activation of platelets, it secrete transforming growth factor beta, TGFB, that will decrease the expression of immune receptor on NK cells, which inhibit their anti-tumor activity. Next is the interaction of CTCs with macrophages. Macrophages express cell surface receptors that interact with ligands expressed by CTCs. For instance, CD47, a receptor expressed by CTCs, interacts with the macrophage surface receptor signal regulatory protein alpha, SIRPα. This interaction inhibits phagocytosis of CTCs by macrophages and allows CTCs to avoid immune surveillance. Once CTCs successfully escape from physical and immune destruction in the circulation, the CTCs will eventually stop when their ability to stick to the endothelial cell becomes stronger than the forces trying to push them along with the blood flow. Then CTCs exit the bloodstream and continue its journey. The second pathway in metastasis is through lymphatic circulation. To understand the lymphatic pathways, we must get to know about lymphangiogenesis. Lymphangiogenesis is the formation of new lymphatic vessels. Tumor-induced lymphangiogenesis contribute to tumor metastasis through lymphatic vessels. For the first step of lymphangiogenesis, we have to know that, on the lymphatic endothelial cell, there is a receptor that is uniquely expressed called VEGFR3, and its growth factor, VEGFC and VEGFD, which differentiated lymphatic endothelium from blood vascular endothelium. VEGF stands for vascular endothelial growth factor, which functions as controlling the growth and sprouting of lymphatic vessels. It becomes the molecular marker to detect tumor metastases via lymph nodes. The growth of primary tumor decreased the diffusion of oxygen within the cells. To obtain the nutrients and oxygen for their survival, tumor cells secrete growth factors VEGFC and VEGFD that promotes the formation of new lymphatic vessels into the peri- and intratumoral areas. The overexpression of VEGFC and VEGFD, and sometimes VEGF and HGF, hepatocyte growth factor, synthesis the sprouting of new lymphatic vessels in the tumor area. This allows the metastasis of the tumor cells into the lymph nodes, and lastly, into the targeted region. Before the CTC starts circulating, VEGF enters the lymphatic vessels and activates the VEGFR3 receptor, leading to increases the density, enlarging and dilating the size. This allows the delivery of circulating tumor cells, thus increasing the incidence of tumor metastasis on pre-existing lymphatic vessels. Next, CTC enters the lymphatic vessels, spreads into the lymph nodes, and serves as major source of lymph angiogenic factors. Post-sentinel lymph nodes undergo remodeling and SMC rearrangement to promote lymph angiogenesis in distant lymph nodes. This leads to secondary metastasis in its preferred tissue or organ. Lymphangiogenesis does not only happen to the primary tumor microenvironment, it also occurred in metastatic tumor-draining lymph nodes. The factors of LN lymphangiogenesis that promote rerouting are most likely the same as tumor-induced. It happens when SLN metastases damage the lymphatic drainage. The lymphatic vessels reroute the flow via collateral vessels. The distal lymph nodes may become false negative SLN, which metastasis-free. Therefore, in later stages, this LN also contains metastatic tumor cells and becomes positive SLN. However, tumor cells may remain dormant in the lymph nodes. Increased lymphatic vessel invasion means an increased risk of CTC metastasis to the distant part and then to the organ, leading to poor prognosis of the patient and death. Last part is the treatment. This therapy works by blocking the immune checkpoint on CTCs, which will activate the immune system to eliminate CTCs in the blood circulation. This therapy used immunotherapy drugs called immune checkpoint inhibitors that target immune checkpoint on the surface of CTCs, such as programmed cell death, ligand-1, PD-L1, and CD47. For example, the immune checkpoint PD-L1 can act as a do not find me signal on CTCs to escape from anti-tumor immune response. So, by blocking PD-L1, programmed cell death ligand-1, it enhances the activity of effector T cells and NK cells in the tumor microenvironment. So these are our references. Thank you for listening and have a nice day.